set now. All right. So we talked about hypertonic, hypotonic, isotonic, and now we're going to pick up from here, and we're going to be talking about the rest of the internal components of the cell. So a couple of quick questions to make sure we're understanding things along the way. Which agents can cause injury to a bacterial plasma membrane? That was mentioned a few slides ago. Does anybody remember from studying? Starts with a Q. Quaternary.
Alcohol.
Yeah, quaternary ammonia compounds. Good. Remember, we have to be careful when we're working in a hospital and using cleaners that are quat cleaners or quaternary ammonia compounds. You have to wear gloves when you use those cleaners because we have plasma membranes too, right? So if I'm hurting the bacteria, I'm hurting myself in that case. How are simple, facilitated, and active diffusion similar, and how are they different? We already covered this one. The simple and facilitated, they both go high to low. The difference is the facilitated needs to use a what? Not ATP, that's going to be active. It needs assistance. It needs a protein channel, right? So it needs a gatekeeper. It needs a protein channel, and then they can move across. The difference then with active is we're moving low to high, and that's where we need that ATP, because we're going up the slide, right? Okay. All right, does this ring a bell? We didn't purge everything from our last exam, did we? I used to do that too. I'd study really hard and then purge it. And be like, wait, what was that? Okay, so now we're moving on in then. So let's talk a little bit about the cytoplasm, and we're going to talk about what else is inside the plasma membrane. So the cytoplasm inside, it's about 80% water, proteins, carbs, and lipids. So about 80% carbs, lipids, protein. And if you picture it in your head, picture kind of an egg white consistency. It's kind of this thick, aqueous, semi-transparent, just like an egg white. And in this region, in this thick goo, we're going to have bacteria have their nucleoid. Remember, that's where they have their DNA. It is similar to a nucleus, but it's not membrane bound. That's the big difference. They have their nucleoid region. They're going to have some ribosomes. And then bacteria are also going to have special compartments that we call inclusions. We'll talk about what an inclusion is. Now, in the nucleoid region, we have that bacterial chromosome that's held in place. And remember, a bacteria chromosome is much simpler than ours. It is long and circular, and it is going to be a simple genetic makeup, meaning that they're just going to have inherited a set of genes from their original cell that produced them. It's not like us where we do sexual reproduction and we inherit two sets of genes, moms and dads, right? So theirs is much simpler. With their DNA then, it can take up about 20% of their internal space. So again, not a huge amount because their DNA is not huge. It's a very small strand. And with their DNA, they can have their main strand, which is circular, like this. So this would be their strand of DNA. But then they can also have some other parts of DNA that's nearby that are referred to as plasmids. A plasmid is small, and it can be separate from the main ring of DNA. A lot of times plasmids will have extra information on them. Sometimes it's like things like how to build a capsule. Sometimes it's things like how to be resistant to antibiotics. Plasmids are helpful for bacteria because these, again, can be shared back and forth. They're really small. These would be what we'll do in their sex P lab. It's kind of like trading baseball cards. They're small, they're easy to trade back and forth. And they recognize which cells have plasmids similar to them and which ones have different ones, and they exchange them. Again, it's like, trading cards. Now, we also can have our ribosomes. We've mentioned that these were something that are found in our eukaryotic cells as well as in our prokaryotic cells. And that these are going to be something that are going to be responsible for protein synthesis. So again, the way this is going to work is we're going to have DNA that's going to come out. We are going to have our DNA be copied to RNA. And then that RNA is going to go to the ribosome and make a protein. And this is the same pattern that we see in our cell process, as well as in bacteria, as well as in plants. We Always do that kind of a process. Now, with the bacteria, the prokaryotes, they tend to have a much different arrangement with their ribosomes, where their components of their ribosomes are a little smaller than ours. This is why we can target them with antibiotics. Because their ribosomes look different, it doesn't hurt our ribosomes. So we separate our bacteria and eukaryotic cells based on the size and arrangement of their ribosomes. So bacteria are going to have a 70S ribosome, and archaea are going to have a 70S ribosome. We'll talk about what that means. Eukaryotes, plants, animals, protists, all of those, are going to have an 80S ribosome. And again, thank heavens for this difference because of the antibiotics. Now, that S that's on there, the S is a unit that we use for the ribosomes, and what it refers to is Spellberg units. Don't you love that? Do you just want to say that name? Spellberg unit. I mean, I just love that name. Named after the person who first developed it. And the Spellberg unit is a measure of settling time. If I mix something in an aqueous solution, how long does it take to settle to the bottom? Okay? So if you look at this, if there's an 80S and a 70S, that means they have a different time for settling. All right? So if I have a shorter settling time, that means I'm a little bit lighter, so I fall, kind of a little bit faster. I'm not having to fight with as much of the aqueous solution, and I can land on the bottom. That's what we see with the bacteria. Now, this gets a little tricky, though. Our ribosomes have two components. There's a large component and a small component that fit together when they're making a protein. Okay, so I have a large and a small unit, and they kind of smash together. Picture like a hamburger bun. A hamburger bun is like two pieces, and when we put them together, we make a hamburger. That's kind of what we're doing here. Okay. Now, my larger unit weighs about 50 Spellbergs. Again, that's its settling time. And my smaller unit is going to be about 30 Spellbergs for prokaryotes. Now, normally, if I took 50 and put it with a 30, I would get 80, right? But that's if we're adding mass. And we're not adding mass. Remember, we're measuring settling. So in this case, it's like new math. 50 plus 30 is going to equal a Spellberg total of 70. Everybody with me on that? The reason we're not adding them together, what we're looking at is their settling time when they're together. All right? So the 50 and the 30, when they come together to make a protein, their combined Spellberg unit is 70. All right? Everybody okay with that? That's for prokaryotes. Eukaryotes are a little different. So this is going to be our pros. Eukaryotes are a little bit different. We have a 60S for our large, and it's shaped a little different, and a 40S for our small. And when these come together, in the perfect world, that would be 100 Spellberg units, right? 60 plus 40, that should be 100. But it's not. Again, we're measuring settling time. So when my 60 and 40 come together, their Spellberg total ends up being 80. This is the difference then between the 80S and the 70S. All right? So I just want to make sure that we're clear on that because I know it's a little odd to kind of picture that. So here's a better drawing than what I just kind of drew on the board. So with my proteins in my ribosomes, in my bacteria and archaea, I have my 50 and my 30 subunits. And together they're going to make a total of a 70 Spellberg. Okay? In my eukaryotes there, I would have the 60 and the 40. They would come together and they would make a total of 80. Yes?
And it's going to stay the same for all.
Exactly. The ribosomes don't change from one prokaryote to the next. That's a great question. So staph has the same kind of ribosomes as bacillus, which has the same as E. coli. It's their same makeup, just like all of us have the same ribosomes in here. So they also have inclusions. And inclusions are easy to think of as like closets. And so an inclusion is going to be something, I don't know, how can you use this so you guys can see better. An inclusion is going to be something that is used as like a storage chamber in a bacterial cell. And basically, some bacteria will store different items depending on what they need to survive. So, for example, some will have a phosphate reserve. And this phosphate reserve is referred to as a metachromatic granule. And what that means is when the bacterium finds extra phosphate, it'll stash it. It'll store it in this little compartment. And then that way, if it needs to have more phosphates for ATP, it has like a little storage area. Again, it's kind of like our little pantry, but it's inside the cell. Some of the bacteria are going to have polysaccharide granules or inclusions where they can store extra starches. We see that a lot of times with like our photosynthetic bacteria because they're making a lot of sunlight and sugar and they'll store a lot of their extras. Some will have lipid inclusions, so they'll have extra fat storage. Some can do CO2 storage, we call those carboxysomes. Some, if they're living in pond water, can store gas in vacuoles, and what they can do is they can inflate or deflate them like a balloon to help them float in the water. Kind of like a fish has a swim bladder to help them float. It's the same idea. They can inhale additional air or shrink it down depending on where they want to be in the water column. And then some are going to be able to store iron in what are called magnetosomes. Again, the inclusions are just little storage regions in the cell where they can store extra things that they need for survival. Now, the reason that not all of the bacteria have these is not all of them need them. For example, if I'm a bacterium that's not doing carbon fixation, I don't need a carboxysome. So they'll have what they need for survival. That's it. They're very light. They're not going to have extra pantries if they don't need them. Now, we've been talking about these in lab. We're going to get a little more detailed here today about endospores. So endospores, as I've mentioned, are going to be kind of a preserve or a resting stage of a bacterial cell. And we've said that endospores are extremely resilient. They are resistant to desiccation. What does that mean? What was that? Moisture, yeah, moisture loss and tearing apart. So desiccation means to lose moisture. So they are resilient or resistant to desiccation. They are also going to be resistant to heat and chemicals. So endospores are very hard to destroy. We mentioned our Clostridium difficile in hospitals and how we worry because those endospores are hard to get rid of. The two main genera that can make endospores are Bacillus and Clostridium. We worked with Bacillus subtilis in our lab, right? But we also have things like Bacillus anthracis out there. We have Clostridium tetani. We have a lot of pathogens that can make endospores. Now, they have a double process that goes with their endospore formation. When a cell starts to realize that maybe it's getting super crowded, it's not getting enough nutrients, or it's starting to get old, it's going to go through what we call the sporulation stage. And what this means is it's going to create a spore inside of it. We said that this is a cell called a sporangium, right, in lab, where you start to make your cell that has the endospore that's visible. That little endospore then is going to be the only thing that stays of that cell. That cell is going to die off. It's going to break apart. Thank you. It's going to die off and break apart. And then what's going to happen is that endospore is going to be left in the environment. And then the endospore, when things are good, it's going to hatch. We refer to that as germination. And it's going to return to what we refer to as a vegetative cell or vegetative state. Now, I know when we think of vegetative states for humans, is that a good thing or a bad thing? Yeah, that's bad, right? When we hear the term vegetative state, we're like, ooh, that means somebody's not really awake or conscious or functioning well on their own. In bacteria land and in archaea land, when you hear vegetative state, that means they're happy and growing. Now, let's think about why this would be. Remember we said before that we used to classify living organisms as flora and fauna. Bacteria were in the flora group, which meant they thought they were plants. So vegetative, when we have a plant producing vegetables, it's happy. So that's why we use the term vegetative. It's kind of a throwback to when we thought our cells were still parts of plants. Okay. I have students always ask me, why do they call it that? So that's why. So with this then, our little endospore will be able to hatch, open up, and create a living cell again. Now here's a picture of an endospore that is forming in the Bacillus anthracis cell here. So again, this is indicating that the cell isn't happy. It's going through sporulation, and it's created this endospore. Now, the process of endospore creation is pretty simple. What they're going to do is they're going to create a replica of their DNA. And instead of dividing like that would normally happen when they create a replica, they're going to take that DNA and they're going to house it inside of a sporulation coat. Now, this isn't reproduction. We want to be careful here. I'm not going from one cell to two cells. What I'm doing is I'm going from one cell, protecting the DNA, and then that cell dies. So I'm not creating additional cells, okay? So I create my little endospore, and to make it super tough, I actually put two membrane coats around the endospore. And that's one of the reasons it's so hard to destroy. We can maybe damage the outside coat, but that inside coat is still intact. It would be like cracking an egg open and there's another shell. And trying to crack that shell open, it's like, really, we have to keep opening this thing. So what's going to happen then is we will go through sporulation, we'll make the endospore inside, and then the endospore will break out of that vegetative cell as it's dying. So as the cell, I should call it a sporangium cell as it's dying. So as the cell is dying and deteriorating, the endospore is released. If we were to come in and look at this on a microscope, just like what we saw in our lab, we saw healthy pink cells, those are vegetatives. We saw some cells with endospores inside, those are sporangiums. And then we saw a lot of endospores just by themselves. The endospores by themselves at one time had been inside of a bacillus cell or had been inside of a vegetative cell. All right? So let's do a quick check. Where is my DNA located inside of a prokaryotic cell? What region? The nucleoid. Good, the nucleoid region. Again, not to be confused with the nucleus, it's the nucleoid. What is the general function of inclusions? If I was to just generally say this is an inclusion and its job is to what?
To store.
Excellent, excellent. Its job is storage. Yeah, and there's a variety of what they can store. But if I just want to be very general, I could say inclusions are used for storage. And then I could get specific and say, okay, yeah, well, this one's used for, storing CO2. And this is, but for now, we're just doing general. Under what conditions do endospores form? What is the cell experiencing when it makes an endospore? Stress. Yeah, it's running out of room. It's running out of nutrients. Excellent. So yeah, the cell isn't happy. So a happy cell is not going to make an endospore. An unhappy cell is going to make an endospore. So our final comparison then, if we did a side-by-side chart, would be something that would look like this with the internal structures. So they are both going to have a plasma membrane, but once we move inside, they can both have cytoplasm. However, things in their cytoplasm can differ. So DNA is in the cytoplasm of the prokaryote in the nucleoid region. Our DNA is in the nucleus of our cells. They have naked DNA, meaning it's exposed. It's not encased in anything. Ours is wrapped with those histones, as we've mentioned several times. It's protected. They can have circular DNA, like a little round circle. Ours is linear, like two lines. They don't have mitochondria. We do. They have 70S ribosomes. We have 80S. They don't have membrane-bound organelles. We do. They have pili that are present. Eukaryotic cells cannot make pili, so we can't do a sex pilus and exchange genetic material. We can't do that. They can have plasmids that are present. We don't have plasmids. Our DNA is all accounted for. It's not in a separate little part to the side. They have a cell wall present. And eukaryotes, we have plants and fungi. that can have cell walls. A few protists, but not animals. And their flagella is made a little bit different. Their flagella is more of a solid, and our flagella is more of a flexible. And what that is because they use flagellin. They use a different protein than what we use. So let's go to the eukaryotic cell and check this one out. The eukaryotic cell, as we know, has additional setups to it, additional layers. So it'll start with that phospholipid bilayer, which we've already covered. But then as we go in, we're going to find that there's a lot more complexity to it. The phospholipid bilayer is going to have a lot of proteins present to help with structure and communication, but we're also going to have a lot of proteins that are inside. So again, just to recap, that plasma membrane is going to allow permeability. It's going to allow diffusion and osmosis. And because our cells don't have a cell wall, we have a lot of flexibility. And we're able to bend, fold, and move our cells in a different way than bacteria. So we can do something called endocytosis. This is where if we come up to a food particle or we come up to some nutrient that we want, our cells can grab and fold and pull it in through the plasma membrane. We can bend around the food particle and then pull it inside, or bend around whatever it is that we want and pull it inside. Bacteria can't because they have that rigid cell wall. If I'm a rigid cell, I can't bend or flex around something. So we have some advantages there. We can also do what's called pinocytosis, and that's where we can fold, grab and fold inward to pull in a lot of water. So let's say we are in a hypertonic situation. Hypertonic meaning that we have lost water because maybe we drank salt water or something. And then what we want to do, remember I told you when I was learning to surf, I drank all that salt water, was super dehydrated. Even though I didn't want additional water, what I had to do was drink water to replace the amounts that I had dehydrated from. And pinocytosis means that what was happening is I was drinking that water is my cells that were shriveled up were gulping the water that was coming in to be able to flush back out. So again, it's being flexible. Bacteria, they just can't do that. Now, if we looked at our cells and we kind of like break them open, we can see that there's some complexity again there. We have the nucleus, which is the large central region of the cell. That's going to be where our DNA is housed. We have all of the little organelles, which are going to have certain functions in the cell. You can think of that as like a company, how a company, when it gets bigger, it'll have, a secretary, it'll have a president, it'll have a vice president. That's kind of like the organelles. They each have their little function or job to help the cell run. We have cytosol. Cytosol is going to include the cytoplasm and all of the compartments inside. The cytoskeleton, that's going to be a structural support made of proteins. And then last but not least, that plasma membrane that is going to control what comes in and out of the cell. So the plasma membrane controls what comes in and out of that cell. Can't get that down any farther. So if we start to look at the structures in our cell, what we see first of all is that cytoplasm as we kind of work our way in from the plasma membrane. And the cytoplasm can also be referred to as the cytosol. And what it is, again, is it's that egg white-like fluid holding everything in place. In there, we also are going to have the cytoskeleton, which are going to be, again, proteins that are going to help support our cells and help them with movement. Bacteria don't have a lot of protein support. And hopefully this kind of makes sense if you think about it. If I'm a bacteria cell, I'm single and by myself in the environment. I don't have to worry about any support to other cells, right? So I can kind of live on my own. I don't have to worry about getting squished or flattened. In our bodies, one of the things I mentioned before, we have cells that are all hooked together, basically. They're all working together, 30 trillion of them. So when I'm standing right now, the cells on the bottom of my feet are like, oh, they're trying to hold up my weight, right? And they yell at me when I eat too many Cheetos. They're like, really, lady? What are you doing here? Like, really? So they're holding up my weight. The way they're able to hold up the weight and not just go squish is because of those special proteins that are inside that cytoskeleton. It's like a scaffolding of a building. If you've ever driven by where they're putting a new building up and you see like the iron structure that's going to be the framework, think of them like that. They're holding things in place. The other thing that cells can do, and this is something that we see with a lot of our eukaryotic organisms like plants, is they can do something called cytoplasmic streaming. Now, the way this works is it means that they can share nutrients from their cytoplasm with one another. Now, if we think about like a plant, it has a leaf, it has a whole bunch of leaves, but it has a leaf maybe getting a lot of sunlight here, and then it has a leaf over here that's in the shade. So this leaf isn't getting as much sunlight, so normally we would think, okay, well that leaf is going to die, and yet it survives. How? Well, the one that's making a lot of sugar from its sunlight shares its nutrients through cytoplasmic streaming to the leaf that's in the shade. So they've got each other's backs. It's like, oh, you need food, I'll give you some food. Oh, you need some phosphates, I'll give you phosphates. And the way they do this is their cytoplasms are able to move within the cell wall, and then they can have components of their cytoplasm move out and cross into the other cell. So it's kind of cool. So they do a lot of sharing and carrying when we look at plant cells. Our cytosol, as I mentioned, is kind of like a jelly-like substance that also helps with holding things in place. And then I mentioned the cytoskeleton is going to be our framework. Now, our cytoskeletons can also help with cell movement. So if you've ever watched, for example, a white blood cell in the body, they can do something called like cell crawling. And it looks like they're a cell just kind of like inching their way along. They don't have flagella, but they're able to move by extending and contracting their cytoskeleton. So they're almost getting like a grappling hook kind of motion, like what we were seeing with the prokaryotes. The only difference is their extension isn't coming out of the cell, it's staying in the cell, but it allows them to do almost like a caterpillar kind of scoot. All right? So white blood cells, they travel or scoot around in our body looking for organisms to attack, looking for bacteria or viruses that might be present. And the way they do that, again, is by extending their proteins and contracting them so that they're able to kind of have movement. Now let's visit the organelles of our eukaryotes. And remember, we do have some eukaryotes that are microscopic. We'll be covering those things like our proteins that are going to have organelles. As we've mentioned before, our ribosomes in eukaryotes are different because they're going to be ADS ribosomes. Now, it turns out that we are going to have ribosomes that can be attached to organelles, and we can also have ribosomes that are just found free and loose in the cell. This means that we can make proteins quickly by whatever ribosome the RNA reaches first. If it's one that's attached, and we'll talk about what it's attached to, it'll make its protein there. If it goes out and hits a loose ribosome in the cytoplasm, it'll make its protein there. It doesn't matter where it makes it, it just matters that it's being read by the ribosome. So we have ribosomes kind of strategically placed all over. Makes things a little more efficient. Now I mentioned that 7DS ribosomes are something that we see in prokaryotes and archaea, but get this. We have 2 organelles associated with eukaryotes that have 7DS ribosomes. That's a little odd, isn't it? I might have to think about that. We have 7DS ribosomes that are present in our mighty mitochondria, in our cells, and plants have them present in their chloroplasts. When scientists found that, they were very intrigued. There's a reason. We'll talk about that in a few slides. Now, if we looked at these cell walls of eukaryotes that are able to make cell walls, so those again would be our plants, our algae, our fungi, we would find that their cell walls look different than our bacteria. They're going to be made primarily of carbohydrates, things like cellulose or chitin. Sometimes they can have glucan or mannan. They're just sugars. They don't have all those protein layers like what we saw inside of our prokaryotes. They also are going to have special structures called glycocalyxes. And I mentioned these before, that these are like those carbohydrate protein signatures that extend from our cells. They were the ones that do the secret handshakes. Remember that? So they help with cell recognition. So a glycocalyx is something that can help our cells recognize one another. We have them in our cells, plants have them in their cells. It allows for recognition. Okay, I am going to take an antibiotic. I'm going to take erythromycin, and my doctor tells me the erythromycin works by binding to the 50S portion of the ribosome. Is this going to affect the prokaryotic cell? Yes, I see nods. Yeah. Remember, our prokaryotes have a 50S. If I take out their ribosome, that means that they can no longer make proteins. And nothing can survive without proteins. Not a single organism can survive without proteins. In fact, viruses, as soon as they get in a cell, one of the first things they start to make are proteins. Okay, so we have to have them. Would it affect a eukaryotic cell? No, because we don't have a 50S. The binding sites on our 60s and 40s are different. So that antibiotic can't bind to our cells. All right, isn't that cool? Cool stuff, right? Now, I did mention, though, that we have 70S ribosomes. Where are our 70S ribosomes? Anybody remember?
17.
Mitochondria, excellent. They're in the mitochondria and in the chloroplast. So you might be looking at this and going, wait a minute, are they going to affect the mitochondria and chloroplast? And the answer is no, because if that antibiotic gets through our plasma membrane, we have other structures that will neutralize it before it can reach the mitochondria or the chloroplast. Phew, we're very lucky, right? Didn't have to worry about that. Okay, let's look a little bit more into the organelles inside of our cells and what they do. So the first big organelle that we can see would be the nucleus. And again, this is going to be what contains our DNA. Think of DNA as kind of like Rapunzel. You guys have heard of the Rapunzel story, the princess locked in the tower kind of a thing? That's how our DNA is. It's locked away in our nucleus. The DNA cannot leave. It's too precious and too valuable, so we always keep it in the nucleus. That's why we also need RNA to read the DNA and leave the nucleus and go to the ribosome. The DNA is not allowed to go out and play. It has to stay in the nucleus. It's too important. We're also going to have some other organelles we'll talk about, which are going to be the ER, that stands for endoplasmic reticulum, part of our transport. The Golgi, which is going to be part of packaging. The lysosome, which is going to be part of digesting and breaking and recycling. And vacuoles, which can again be used kind of as storage units or like pantries inside of our cells. So let's take a look at these. We also have the mitochondria that can help with cell respiration. That means creating ATP. In some cells, we can have chloroplasts that will help with photosynthesis. Unfortunately, we don't have that ability. Wouldn't you love that? Like go sit out in the sun and then get energy? Oh, that would be awesome. But unfortunately, we don't have chloroplasts. And then peroxisomes. Peroxisomes are going to be special organelles that help neutralize toxic forms of oxygen. Oxygen, as we know, and we've mentioned in here several times, it's an electron hog. It likes to pull electrons towards it. And sometimes it pulls so many that it gets super negative. We call that like a superoxide. And our cells have to be like, okay, oxygen, calm down. You need to calm down because you're getting toxic. And that's what our peroxisomes do. They release nutrients that help the oxygen calm down. They're like, you need to just chill out. So we're going to talk about those and how they help our oxygen relax again. If we were to look at our general cell, this kind of shows you what the main functions of each of the organelles are. So the nucleus, you can think of it as like the control center. The cytoskeletons are going to be like for structure. Powerhouse organelle is going to be mitochondria. The lysosomes are like a cleanup crew. Plasma membrane is like a bouncer. Golgi's distribution. Ribosomes are the workbenches where we make things. So let's look at a little bit with the rough endoplasmic reticulum. So this is showing you that the rough ER is going to be found right outside of the nucleus. So it's highlighted there in green. So it's attached to the nucleus right on the outside. And what we're going to see is that the rough ER is referred to as being rough because it has ribosomes embedded in it. And this is going to be where we can make most of our proteins. Now, hopefully it makes sense that our rough ER would be right outside of the nucleus. Because remember, the DNA is in the nucleus with the information about what proteins to make. So the RNA reads the DNA, leaves the nucleus, and immediately hits a ribosome. And that way we can get proteins made quickly. So it's very efficient to have them like right there. Now, as I said, we can also have ribosomes that are loose. They can also just be free in the cytosol, but the majority of them are found in that rough ER. That's what we're going to focus on. And the protein that we're going to make, it can be made on the interior of the rough ER, and what that means is closest to the nucleus. So we're making proteins right beside where that nucleus is. Now, in that endoplasmic reticulum, or in the ER, we started by having the rough ER, that's full of bumps, that's where we're making our proteins. But if I keep moving out of that membrane, I hit an area where the rough ER starts to become smooth. And the smooth ER is going to be a part of that membrane that is not processing proteins, but instead is processing lipids and fats and detoxifying harmful substances that the cell has been exposed to. So for example, when we have a holiday celebration or Super Bowl Sunday, maybe we drink a little too much, right? We drink a little alcohol. Maybe after our exam on Monday, we drink a little much, right? So we have some alcohol come into our bodies. That alcohol is considered to be a toxin by our cells. So what they're going to do is they're going to send it to the liver, and the liver is going to neutralize the toxin. So the alcohol is going to go to the liver. The liver cells are going to absorb the alcohol, and they're going to move it to the smooth endoplasmic reticulum. And the smooth ER is going to help neutralize or break the alcohol down into its components and make it harmless. Now, where we run into trouble, alcohol has a lot of sugar in it. What happens if you drink too much alcohol? You get a hangover. Yeah, you can get dehydrated. Good. That's good. The hangover is actually from dehydration. If you're drinking, drink water, and you won't have as much of a hangover. See, I'll give you life lessons here. You don't want to have a hangover, just constantly drink water. Use it as your chaser, okay? All right. I sound like I know what I'm doing up here, don't I? Yeah. Okay, you guys are all like, yeah, she's so slush. Okay. All right, so anyway. All right, whatever. Okay, so we're going to have our cells that are processing the alcohol. All right. The other thing that we run into, though, have you ever seen people that have beer bellies? Yeah, what's up with the beer belly? It's because there's a lot of sugar in the alcohol. And so what happens is our liver cells get rid of the harmful stuff, but when they break alcohol down, there's a lot of sugar in it. And so that's why they tell you if you're trying to lose weight, don't drink as much alcohol. It's because there's a lot of sugar stored in there. And it's a lot that your body can't process at once, so your body goes, don't you worry, I'll store it for later. And so it turns it into a fat resource. So that's why we have to be careful. Don't you love how our bodies always have our backs? They're like, don't worry, I'll store it. It's like pigs. All right. The other thing it can also do is it can also make hormones in this region. So the smooth ER is also where we're going to be involved with hormone creation. This is what our ER looks like. Oh, yes, go ahead.
So, for the sugar to be stored, does it also go to the smooth endoplasmic reticulum before it goes and gets stored as sugar?
Yes. In the smooth ER, that's where the smooth ER kind of sorts everything out, and it goes, this is a toxin. Oh, sugar, we'll store that. And then that sugar will be packaged and moved elsewhere. You got it. Great question. So here's what our endoplasmic reticulum looks like, our ER. At the top, which would be closest to the nucleus, we have our rough ER, where all the ribosomes are, and that's where we're making all of our proteins. As I move out of that rough ER, I start to hit a lot of folds, and we call that the smooth ER. Again, that's where we're going to process lipids, toxins, and hormones. So lipids, toxins, and hormones. Here's a picture of what it looks like under an electron microscope. So you can see the rough ER has all these little bumps in it. Those are all those little circles. Those are going to be all the rough ER, the ribosomes. So all of these little circles that are in here are the ribosomes. And then I move out and I see just a lot of folds. And all those folds are indicating the smooth ER. Now, let's say I've made my protein in the rough ER, or I made my lipid in the smooth ER. That's great, but now what am I going to do? Or maybe I broke down my alcohol and I have sugar. That's great, but now what am I going to do? I have to go to the next step in my process, which is going to involve the next organelle. The next organelle we're going to involve is something referred to as a Golgi body. It also used to be called the Golgi apparatus. You might have heard that before. Golgi bodies are basically little stacks of membranes that are going to be involved in packaging whatever the cell has manufactured. So think of the Golgi as kind of like a UPS or Amazon. If I order something from UPS or Amazon, they take my item, they put it in a box, they tape it up, and they send it to me. That's kind of what's happened here. My ribosomes have made a protein, or my smoothie has made a lipid. It moves to the Golgi, and the Golgi is going to package it up, tape it up, and send it to wherever it needs to go. It's our packaging process. So when we package, we're going to put it in a little membrane that we call a vesicle. And this little vesicle then is going to have information in it that tells it where to go, just like how on our packages we have our addresses written. It tells the cell whether it's going to stay inside the cell, whether it's going to be sent somewhere else. It tells the cell where to process. And then this little package is ready to go out and do whatever job it needs to do. So here's a picture of how that works. So we've come out of the rough ER, we've come out of the smooth ER. The next thing we hit is that peach or pink area that's all folded. That's the Golgi. The Golgi is going to take what we made. It's going to put it inside of a vesicle. The vesicle are these little cup-like structures that are nearby. You can see it here and here. And these little cup-like structures can then move out. They can leave the cell. They can stay in the cell. Or they can be moved to a recycling organelle where things that we don't need anymore can get broken down. So we're going to have these little structures then that are packaged that move around. Now, one of the things that our cell will have happen a lot is we'll get things that we don't need anymore. Kind of like that alcohol, we break it down, we don't need it anymore. If we have used a food particle and we've taken all the nutrients we can from it and we have some leftover stuff we don't need, or if we have a bacterial cell invader and we can grab and capture it, we're going to send all of the things we don't need or things that we want to kill to a special organelle called the lysosome. The lysosome is like our recycling center. It is a little organelle that is full of caustic, acidic material. And so a vesicle will come up to it. Maybe it contains inside some leftover protein that we don't need. It'll merge to the lysosome and release its internal components. And the lysosome, which is very caustic, will break down what's present. So it's kind of like a little trash recycling, or a little center that like churns things up and gets rid of them. Yeah.
So going back to the Golgi, do all the proteins have like a vesicle around them as they go through?
So yes, when they're processed by the Golgi, they get packaged in a vesicle. Yep.
No matter where their destination.
Exactly. Even if it's inside of the cell, because the Golgi helps us know where it goes. It's like able to read that part of the ribosome and it can tell if it needs to stay interior or if it needs to go exterior. Great question. So what's going to happen then with these lysosomes is they're going to help us with digesting and recycling. Now, what's interesting is the lysosomes can also help with cell destruction. If a cell starts functioning in an odd fashion, or if a cell gets too old, the DNA will make a protein that's like a death protein and send it to the lysosome. It's like a signal. And the lysosome will get this signal from the DNA and it will actually burst or rupture open. And it releases all of the caustic material inside of the cell. And what that does is it destroys the interior part of the cell and most of the plasma membrane, but it can't cross. It can't leave the cell. So it's going to cause the cell to basically deteriorate, break down, and then the plasma membrane that's left behind kind of withers and fades. It's basically a programmed cell death. So lysosomes a lot of times are also referred to as suicide sacs. They're ways for our cells to get rid of a cell that's not functioning correctly. So if we detect that maybe a cell is starting to do too much reproduction, that could lead maybe to early cancer. We'll talk about how that works. But our cells, if they can catch it soon enough, they're going to destroy it. They're going to try to get that cell out of there. So they're always regulating one another to make sure the cells are performing okay. All right, let's check our understanding here. Compare the function of the nucleus of a eukaryote to the nucleoid of a prokaryote. So how are they similar? What do they have? They have the DNA, yep. And what is different about a nucleus and a nucleoid? The nucleus has a membrane, right? The nucleus has a membrane and the nucleoid doesn't. So the nucleoid is a general region, but it doesn't have a membrane. It's not like a sac. The nucleus is like a sac. How do the rough and smooth ER compare structurally and functionally? So let's break this apart. What makes the rough ER rough?
Ribosomes.
Ribosomes, excellent. So what is made in the rough ER? Proteins, excellent, yes. So the rough ER is going to make proteins. In the smooth ER, I'm smooth because I don't have ribosomes, but I'm going to make other things. What does the smooth ER make? There's a couple of things. If you give me one. Yeah.
Lipids.
Lipids, excellent. It's going to make lipids.
Hormones.
Hormones, excellent. It also makes hormones. And then there's one other thing it helps with. Detoxifying. So yeah, detoxifying, excellent. So all of those things that are very important happen in the smooth ER. Now, one of the most important organelles that we have is the mighty mitochondria. The mighty, oh yeah, go ahead.
Can you damage those hormones like
They can. If your body is experiencing too much heat, or if your body is experiencing too much acidity inside, hormones are basically a protein structure and a lipid structure, and so they can be denatured. So hormones can be affected. We see this, for example, with certain athletes. Think of like our gymnasts. Have you ever noticed that gymnasts are very, very small?
Yeah.
One of the reasons they're really small is they're training all the time, so they're using lots of energy, but they also tend to reduce their metabolic input, meaning they're not going to take in as much food when they're starting to grow. And part of that is usually by choice. They're training so much and they want to not gain weight. The problem with that is that can affect hormone production. This is why a lot of times women who have a very low weight will not menstruate. It's because it affects their hormones, it shuts them off. So with a lot of our gymnasts, for example, we see delayed body formation because they're not producing hormones, and if their body tries, they just don't have enough nutrients present, or the hormone will get damaged because of all of the intense training and workouts. So it's kind of an interesting thing that we see in the low body size.
Does that affect like menopause in their later years? Does it come on faster?
It does affect, actually it can even cause it to come earlier sometimes. So menopause a lot of times can be triggered by stress. If our bodies are stressed, they can go through an early menopause. So it's usually, you're right, it's usually based on when you started your menses or menstruation to when you end. But a lot of times with stress or other factors, that can affect that as well. Good question. So our mighty mitochondria is something that's fascinating for us because it's going to be the organelle in charge of making energy. It's going to be where we make our ATP, most of our ATP. So this is going to be one of those organelles that's very special because it has 70S ribosomes and it even has its own DNA. This was another thing that shocked scientists. We looked at this and we're like, what is going on here with this organelle? It's a very strange one. And there's a reason again. Now, the mitochondria, what's interesting, our cells can make mitochondria depending on our energy demands. If you have not worked out a lot, like if you're like me and you work out only when you have to, okay? I'm not a gym rat, I'm the first to admit that. So I don't like to work out a lot. My cells probably have like 2 mitochondria. Okay, they probably have more than that, but still, like they don't have a lot. My friend Kelly, who is like a total gym workout person, she runs every morning. I'm so amazed by her. Her cells are full of mitochondria. So if you're an athlete or if you work out a lot, your cells will make extra mitochondria to meet your energy demands. This is why when you start a new exercise regime, a lot of times you feel tired. You're putting stress on your cells because you're asking for extra energy and they don't have it yet. But your body is so easy to, change. It'll be like, okay, are we going to need energy? I'll make more mitochondria. And it will send a signal to the mitochondria telling them to divide and replicate, and we'll see the mitochondrial numbers increase in our cells. Yeah.
Is that like something that shows you like sometimes like you said like sometimes you work out and you feel like you're running but then you stop but then you're not tired and you go for another one?
Right, you keep going, yeah, you get used to being like, okay, I'm gonna do a 30-minute workout, but as your body builds up, you're like, well, I can do now 45 or an hour. It's because you have more ATP available. Exactly right. There's also that thing though where you can use too much. Have you ever heard hitting the wall? Where you like work out and then you just feel like you're gonna pass out or you get dizzy? It's because you've used all of your reserves. And your body's like, whoa, we need to sit down for a minute. You know, maybe have a cheeseburger, get some protein in here because something's going on, right? Maybe not a cheeseburger, but you get the idea. Okay, so our mighty mitochondria then have their 70S ribosomes, their own DNA, and when we got the ability to use scanning electron microscopes and see what this organelle looked like, we were shocked. Look at this organelle. This organelle looks very bacterial in nature. It looks like a prokaryote. It looks like a bacillus of some sort. What we saw is there's lots of folding in there. There's these like cristae, those are folds, there's these membranes, and we were like, wait a minute, that almost looks like a bacterial cell inside of our bodies. And sure enough, that's what it turns out being. There's a theory out there called the theory of endosymbiosis. And remember, theory means lots of proof behind it. So endosymbiosis states that many, many years ago, like millions of years ago, our organelles started to evolve in early eukaryotic cells. And we think the first eukaryotic cells, our ancestor cells, were single-celled. Okay, so they were just trying to survive. And they would eat anything that was in the environment, and what we suspect is that a eukaryotic cell ate or consumed a prokaryote. It was going to break it down for food. But instead, that prokaryote, instead of being neutralized in the cell, it was a mass energy producer. It was just a random type of bacteria that can make a lot of ATP. And it started to produce a lot of ATP in the eukaryote. And the eukaryote went, oh, this is good. I'm getting a lot of energy from this little thing I swallowed. I'm not going to digest it. And so they started living together. We call that symbiosis. They started living together. And so the reason they call it endosymbiosis is because the mitochondria went inside. Okay, so endosymbiosis, meaning they're living together inside. So then what's fascinating is when our eukaryotic ancestor divided, the mitochondria inside divided too. And that meant that its offspring now had a mitochondria. And this kept happening. As our eukaryotes started to develop, and we think part of the reason they were able to develop is because of the mitochondria being present. They were able to become multicellular because they could make more energy now. Isn't that cool? Because of this little bacterial cell that we happened to pull in, it led to multicellular organisms. Now, the other interesting thing about our mitochondria then, they have their own DNA, so they can divide on their own. They have their 70S ribosomes, which shows that they were bacterial in nature. They also are something that we only inherit from our mothers. So if you look at sperm cells, there is not a single mitochondria present in that DNA information or in the sperm cell. If you look at an egg cell, there is DNA information and a mitochondrial present. So when you are born, our mitochondria are present are from our maternal lines. So think about all of our ancestors that are inside of our cells right now. Isn't that kind of a cool thing? I mean, they're going back like great-great-great-great-great-grandmothers. You know, we've inherited their mitochondria. So it's kind of neat to think about the fact that you're carrying your ancestors with you right now. We think the same thing happened with a chloroplast, except that eukaryotic line that ingested the chloroplast gave them the ability to break down sunlight, and that led to the evolution of plants. So animal cells ingested a bacterium that was the mitochondria. The plant cells ingested a mitochondria and then later ingested a chloroplast, so they have both. And they're able to do sunlight as well as ATP. So over time then, these became integrated inside of us. If we try to separate the mitochondria from our cell now, it can't survive. And our cell can't survive without it either. They are completely attached to each other. We don't make enough energy on our own. to not have a mitochondria. But if we pull the mitochondria out, it's not protected or taken care of, and it dies. So it can't make it either. So we are completely reliant on each other. The fact that this happened was probably like a 1 in a billion odds, but it worked beautifully for the evolution of life. So with our mighty mitochondria then, they're going to be harvesting energy. They have double membranes that are similar to what we see with bacteria. They're the same size as modern bacteria. They have these folds that we see with bacteria. Those folds are kind of like the internal storage units that we talked about with the bacteria, same kind of idea. They are the same size. They have ribosomes that are present that are 7DS. And again, they're going to divide. I might have to speed this up a little bit. They're going to divide by fission, which is like modern bacteria. So that means they're able to just kind of separate or split in half. It's a very simple process for reproduction. So we have learned that mitochondria respond to our environment. So this is why a lot of times athletes will go and train at high elevations. One of the things mitochondria need to make ATP is oxygen. So if I go to a high elevation and train, is there a lot of oxygen at that high elevation? No. So what happens is my body goes, oh, there's not a lot of oxygen here. I should make more mitochondria, and then they can process the little oxygen that's present faster. So their cells make a whole bunch of mitochondria, even more than if they were at sea level. And then when they come back to sea level, they have all this extra energy.
Is that a good thing? Like,
It is. It doesn't hurt us at all. It doesn't hurt us at all.
So,
Yeah. What about? Sorry. You want to go?
I was just gonna add, like,
Yeah.
so for example, like people from Peru or those high elevation,
Yes.
like I'm sure that benefits them.
Yes, yes. So they have a lot more energy. They're able to hike farther. They're able to lift more loads. It's because of the fact that they have more mitochondria present in their cells.
Yeah.
Yeah.
What about for like is it vents for scuba divers? How does that?
So that's different. That's when you get nitrogen bubbles.
Okay.
So that's when we go in the water, our cells start to decompress from the pressure. So we get kind of pushed in. The oxygen that's normally in the cell gets pushed out. And then what can happen is when we're breathing in through scuba tanks, one of the gases that's in a scuba tank is nitrogen, and then nitrogen starts to build up. When you're surfacing, you need to slowly surface and breathe out to breathe out that nitrogen and replace it slowly with oxygen from the tank as well. We're trying to keep it in balance. So our cells are decompressed. As we slowly rise, we need to make sure we're getting enough oxygen to help them push out. If you come up too fast, you're only going to have a high number of nitrogen and a low number of oxygen, which causes pain in the cells. So that's what the bends are. It's because it's causing cramping. Your body needs more oxygen in its stat. Usually the treatment is they'll put you in an oxygen chamber. So you're breathing in pure oxygen to replace it as fast as possible. So we have a lot of evidence then with our mitochondria. If we were to look then at our cells side by side, we have two of our most common eukaryotic cells here. We have the animal cell and the plant cell. The differences are shown in red. So the animal cell is not going to have a cell wall. The plant cell is. So that's why it's shown in red. We have a special organelle called a centriole. That's the one shown in red there. The next one is in red. And the centriole is going to help us with a process called mitosis, which is cell division. Plant cells, they don't have centrioles. We're something that's specialized to us. They're going to have chloroplasts that allow photosynthesis. Unfortunately, we don't have those. So those are the big differences, but everything else is going to be the same. That down. Everything else is going to be the same between the two. All right, so that gets us through our cells. Let me get out of here. And now, what's that? Except for the nucleus, that's right. And now we're going to jump and we're going to go to our next chapter, which is going to be metabolism. Yeah, what you got? So our bodies can handle a little stress. We want to be careful though. We want to make sure that the stress that we are experiencing is one that we can recover from. So bodies can handle a temporary amount of stress, like let's say I go to a higher elevation, and they can adapt. But if I have a lot of external stress, like a lot of pressures or abuse or things like that that are going on, it's harder for the body to adapt. So I need to make sure that I'm taking in nutrients, I'm taking in water, I have lots of oxygen. Like I have to make sure that I'm meeting other needs to help it adapt. But when you're working out, you are stressing your body in a good way. You're increasing your heart rate, you're pushing your body to make more mitochondria, and that's why if you do a workout, it's healthy. But can you work out too much? Can you go too far? Yeah, you can go too far. You can have people that go too far and they actually damage their muscles or they'll damage their body. So it's, again, it's all about moderation. Just like with lipids, it's all about moderation. Can you have a donut? Yes, you can have a donut. Can you have a donut every hour? No, don't have a donut every hour. It's all about moderation. Okay, it's a balance. I know, it's like we have to learn the balance, right? Okay, even though we want a donut every hour. Okay, so here we go, lecture 5. We're going to talk about metabolism. So I love this because one of my cousins went vegan on us, and we love her dearly, but it's like, it's so hard planning family meals now, because we'll do like, a barbecue, and we're like, wait, she can't eat that. Okay, wait, can she eat eggs? Are you allowed to eat eggs? We're like texting her, eggs? No, I don't eat eggs. Oh gosh, okay, what are we going to feed her? So it's like this constant struggle, she's like 15, and we're like, we don't know what to give her. It's like, here's a pixie stick with sugar, eat that. It's like, we don't know. We're figuring it out. And she's so funny. She won't tell us either what she likes to eat. She likes to watch us like stress, Yeah, it's 15-year-olds. So anyway, so I love this comic because it just made me laugh. It reminded me of my little cousin. Okay, so let's talk about metabolism. So you've probably seen metabolism in a few of your other classes, like physiology. Maybe you still have 9 years from it. Metabolism can be pretty complicated. It's something that it's easier to kind of learn a little piece at a time. Now, there are hundreds of metabolic pathways that our bodies can follow. And I'll try to remember to show you a picture of a metabolic chart. You might have seen them. It basically looks like a roadmap gone wrong. It's like arrows everywhere, lines everywhere. It's like, oh my gosh, all the ways that our bodies can process. We're going to follow one metabolic pathway, the one that creates ATP. So we're going to follow glycolysis, Krebs, and the electron transport chain. Those are going to be the ones we're going to follow.
Yeah, she's supposed to be still on the class. I don't know if she's able to follow you still.
Brianna? Yeah, she can still see me.
Oh, okay.
Yeah, it's switched when I reopened it because it's a newer PowerPoint. It doesn't show.
I thought maybe she got knocked off.
Yeah, no, she's still good. Brianna, you can still see me, right? I have you in the chat.
Sorry, I didn't mean
No,
to
thank you. Oh, yeah, I got a thumbs up. Nope, we're good. Thank you. Thank you for letting me know. I appreciate that. The difference is my other one was an older PowerPoint, and so it wasn't converting over properly with my computer. I'm just letting it go. It's me and technology. I always have issues. Okay, so our metabolism then, it's going to be used to help digest and break down things like the carbohydrates we eat, the fats we eat, the proteins we eat, and the nucleic acids we eat. Wait a minute. Do we eat nucleic acids? Do we eat DNA and RNA? We do. When you eat lettuce or spinach, what's inside of there? DNA and RNA. We're eating leaves. We're eating cells. When we eat steak, inside of that steak is cow cells. So we get DNA and RNA from our diet. And what our body does is it breaks it down and rebuilds it to what we need. Okay? So again, it's like me handing you a Lego car, and our body gets the Lego car, breaks it down, and rebuilds it as a plane. Weird stuff, right? We don't think about this, right? You want to know another one that'll blow your mind? When you, no, I don't want to know anymore. When you eat fruits, fruits are ovaries.
What?
Yeah, I know, we don't think about it that way. Yeah. So when you eat a fruit, that fleshy part around the seed is basically like the ovary that we have that protects our embryos. It's a plant ovary. And so when it develops, it's a tissue that's supposed to support or nutrient, take care of the seed inside. So what do? We eat it and then we throw the seed away. Isn't that horrible? We're murdering a plant, right? My little cousin who's a vegan would be all upset about this. Oh, it's so horrible. Yeah, I think this is why she doesn't eat things anymore. She's like, I don't know what to eat anymore because I don't want to eat an ovary. Yeah, it's that kind of thing. But it's also kind of cool because it's like a circle of life thing. So why do plants want us to eat fruit? Well, the idea is that if an animal eats the fruit, especially if it's like a horse or a cow, they eat the seed too. And then they travel off, and then eventually when it defecates out, it's perfect. The seed has fertilizer, It's in a new location where hopefully there's not a lot of competition, and boom, I can develop a new apple tree, or I can develop a new plum tree. It's beautiful. Isn't that great? We kind of mess it up a little. We throw the seed away, but the seed goes to the dump, and guess what? It can grow there, right? So if it goes to an area in the dump, it can grow there. So we'll think about that. Or your compost pile, it can grow there. Okay. So the idea then is metabolism is taking in nutrients, breaking them down, and rebuilding them. We have to have certain types of metabolic products to be able to survive. If we stop eating, we can live about 14 days, and then that's about it, because our body runs out of nutrients. We have a little stockpile inside, but after about 14 days, we're done. We're also going to find that for a lot of metabolic systems, oxygen is essential. We need it for our metabolism. This is why if we get into a situation where our oxygen supply is cut off, we can die. It's because we're not able to create additional ATP or proteins or nutrients. And so if we're asphyxiating, that means our brain is no longer getting oxygen and it no longer has enough energy to survive. And we can go for a few minutes, and then that's about it, about 5 minutes before you get some brain damage. We also are going to have some bacteria that are going to be obligate aerobes. And what that means is, like us, they need oxygen to survive. One of them is Mycobacterium tuberculosis. Where do TB infections occur in our bodies? When you picture TB, what do you picture? Lungs, coughing, yeah. The reason they're in our lungs is they need oxygen to survive. They wouldn't make it in our digestive system. They wouldn't make it in our deep tissue. They have to be where there's nutrients and oxygen. We're going to find out that we also have some, though, where oxygen is toxic to them. We call those anaerobes. Anaerobes are ones that live without oxygen, and in some cases, the oxygen is so toxic, it'll kill them on contact. This is what we see with our Clostridium group. We talked about Clostridium tetani, and I told you it makes spores. If you picture Clostridium tetani, it's in our environment, it's out in our soils, it's all around us. We don't worry about it, though, because we have all this beautiful oxygen around us, too, which kills it and keeps it from growing. So right now, all the Clostridium around us is in a spore state. Where we worry is when we get a puncture wound. If we get a puncture that pushes into the tissue, pushes into the deep tissue, which is anaerobic, even if I pull that nail out or whatever punctured out, I now have endospores inside in an anaerobic tissue. Perfect for them to start growing. That's why we worry about getting a puncture with Clostridium. Same with Clostridium difficile. When it's in a hospital room, we worry, but we also know that most of our patients will be fine because of all the oxygen. It's if they consume the endospores, then we get worried, because then the endospores can move through and potentially hatch out, especially if they're immunocompromised and don't have a strong normal flora to keep the C. difficile from growing. So we're going to find that some of our bacteria are going to require oxygen and others are going to hide from it. So metabolism in general is going to be the sum of all of the chemical reactions that occur in an organism. So creating proteins, creating lipids, creating nucleic acids, that's all a part of my metabolism. And with my metabolism, I'm going to get energy movement and energy flow. So we're going to see that energy can be transferred from one molecule to another. And we refer to these a lot of times as redox reactions. You might have heard of redox reactions. Redox reactions is where we are going to have electrons moving from one molecule to another based on the pull of that molecule. Now, the way to picture this, when we picture redox reactions, picture I have four of you holding the corner of a sheet, okay? And in the middle of the sheet, I put a tennis ball, all right? That tennis ball is going to cause the sheet to go down a little bit. That means that tennis ball has a little bit of an electron pull, okay? So it's going to pull down a little, has a little electron pull. And now while you guys are standing there chit-chatting, I wait till you're relaxed, and then I throw a bowling ball on the sheet. I know, isn't that mean? What's the bowling ball going to do? It's going to really pull the sheet down. And the little tennis ball that's there is going to pull into the bowling ball. If that tennis ball had any electrons, guess where they're going to go now? To the bowling ball, okay? So the idea behind this is we're going to get electron flow or electron movement based on the weight and the pull of molecules. All right, so that's going to be one of the things we're going to see. So we're going to eat food, it's going to come in, and electrons are going to flow or be pulled to other molecules because of their attractions and the fact that they're big. It's like a bowling ball to a tennis ball. All right, so we call these redox reactions. Now, the way you can think of which way the electrons are flowing, if I tell you that there is an organism that has had oxidation occur, oxidation is the removal of electrons. A way to keep this in mind is with the acronym LEO-GR. Have you guys ever heard that one? LEO-GR. What animal do you picture when you hear Leo? Lion, yeah, so LEO, Leo. So loses electrons, that's the LE, it becomes oxidized, or oxidation. Okay, so Leo. GR gains electrons, it becomes reduced. All right. There's another one you might have heard. Have you guys heard of oil rig?
Yeah.
Yeah, that one's one they use a lot of times in chemistry. So it's whichever one you like. Oil rig is another one, which is oxidation is losing, rig is reduction is gaining. Pick whichever one you like and learn that one, whichever one you like. Those are just little acronyms. Acronyms are helpful to help remember flow or to help remember how things are moving. So LEO-GR loses electrons oxidized, GR gains electrons and reduced. Oil rig is oxidation is losing, reduction is gaining. Yes?
So would this coincide with ionic, the electron transfer?
Very similar. So remember when we were talking about like covalent bonds and we said the oxygen would hog the electrons? Similar idea. We're just going to have molecules that are going to have maybe more oxygen, so they're going to pull electrons towards them. Or molecules that have more negativity, they're going to pull electrons towards them. So yes, you're thinking the right lines. Okay? So with this then, the other thing we need to think of is with oxidation and reduction, they always happen together. I can't oxidize something without reducing it, meaning that if I'm losing an electron, it has to go somewhere. So I have to have something gaining it. Now, one thing that can be confusing is this idea of reduction. We can have reduced forms of certain materials. For example, carbons can become reduced. Methanes can become reduced. And what that means, remember, is that means that they are pulling electrons towards them. So gaining electrons means I'm getting reduced. Now, the way that this works then is we're able to kind of manipulate who they're bonding with, and then they will pull electrons to fill those bonds. Just like before when we removed water, remember when we pulled water out, we'd free up the carbon end, and then it would pull another molecule near it? That's what we're doing here. Okay, we're freeing up carbons that can then pull electrons or molecules towards it. They could also have some that will lose electrons, and these are going to be things like aldehydes, ketones. These are things that we have to be careful of in our bodies, right? If we hear that we have too many ketones built up, we have to be careful with that, because what that means is they don't have a lot of energy available, and if anything, what can happen is that they can lose electrons, and once those electrons are lost, they become a dead particle. They don't really do anything for the body. So we have to be careful. This is why diabetics have to watch for ketoacidosis. We don't want to have too many ketones build up, because what it does is it takes up room in the body and there's not a lot of energy available. Okay, so we have to be careful with our ketones. Now, I often hear people say, okay, well, diabetics have to watch ketoacidosis, but we want to have ketones when we're doing low carb. Like, what's the difference? Ketoacidosis is a ketone that has an acidic quality. It's lost its electrons, and it is no longer able to provide energy. That means that the diabetic person is no longer going to be able to process energy well. A ketone from eating low carb is something that you're replenishing constantly with additional low carbs. And so those ketones can continue to lose electrons and be cycled through because we're replenishing. Okay, so it's a little bit different. Okay, I think we'll stop here because I'm seeing everybody wants to go study. So we'll stop here. We'll pick up to you next time. Yes, sir.